This is Chapter 3, Part D, Cell Life Cycle, Protein Synthesis. So, the cell undergoes a life cycle. It basically is just what it sounds like. It's a life cycle. It is the changes from the formation of the cell until it reproduces. Most of the time the cell is in a phase called interphase, and then it enters this part of the cellular division phase called the mitotic phase. Now, some cells are amitotic. They don't ever go into the mitotic phase. They stay in interphase their entire existence. Things like neurons would fall into this category. Skeletal muscle cells fall into this category. They don't reproduce. Other cells reproduce, but they do it slowly. They do it over a course of a long time, like hepatocytes and things like that. And other cells, such as skin cells, reproduce very, very quickly. So interphase is punctuated by a lot of mitotic phases. So there's a mitotic phase, a new cell is born, it goes into interphase and then enters its own mitotic phase and divides again. So interphase is really the phase in which the cell is doing the things that that cell is supposed to do. So it's growing, it's having its functions happen, all the chemical reactions, all the protein synthesis, all of that happens during interphase. During the mitotic phase, none of it really happens. So this is the period from the time the cell is formed until it's divided. The nuclear material, all the DNA, is found as chromatin. It's found unraveled off its spools. And there are four subphases of interphase. Again, depending on the type of cell, but generally speaking, there are four types. So it's basically G1, S, and G2, or G0 if they permanently cease to divide. So what happens in the case of, say, a neuron is that it just stays stuck in G0. So we find G1, S, and G2 in mitotic cells, and we find G0 in amitotic cells. So I don't quite know how to get this across, except to just say it. During all of interphase, the cytoplasm is undergoing the chemical reactions that are appropriate to that cell. So if that cell is a cell in the adrenal medulla, it is making norepinephrine. If it's a cell in a goblet cell in mucosal membrane, then it is producing mucin, the protein mucin, to make mucus, etc. If it's hepatocyte, it may be producing bile salts, something like that. So this happens throughout the interphase. In G1, the cell is growing and is actively metabolizing and doing the job of the cell. The only difference between G1 and S is that during S, all the cytoplasm is still doing the same crap that it was doing in G1, but the difference is that in the nucleus, the chromatin starts to replicate. So the DNA that's contained on the chromatin makes copies of itself. As far as the cellular activities are going, they are virtually uninterrupted. So S starts with the beginning of DNA replication, and S ends with the ending of DNA replication. Once all the DNA is replicated, then S is over. G2, it says, is preparing for division, but really G2 is just more growth and metabolism and more of the same, doing the work of the cell. The difference being that now there are 2 copies of every strand of chromatin in the nucleus of that cell. When we look at this picture, and then you look in your book that gives you time frame for these, please don't pay attention to the time frames that closely. That's for an epithelial cell. These things can vary wildly between different cell populations. So G1 is growth or a gap. I prefer the term growth because it is more indicative of what's going on. The cell is growing, it's getting bigger, and it reaches a certain size and a certain level of metabolism. The DNA is replicated and synthesized in the nucleus, but there's still more growth going on. And in G2, there's still more growth going on and more metabolism going on, but the DNA is already replicated. In the mitotic phase, which is the rest of it, there's 2 parts. There is mitosis, which is this. It is prophase, metaphase, anaphase, and telophase, and that is the dividing of the nucleus. Mitosis is not cell division. Mitosis is nucleus replication, nucleus division. Cytokinesis is the other part, and it kind of starts about here and gets really rolling after telophase, after the second nucleus is formed, and that is the actual cytoplasm division. So, so during interphase, the chromatin is all over the place, and all of the, the myofilaments and microtubules and all of that stuff is all throughout the cell. The chromatin is all unwound and being used for RNA synthesis, for transcription purposes in making proteins. Now, what happens here in the chromatin, on each of the strands of the chromatin, the DNA starts to unwind, and as it untwists, the 2 parts of it zip apart, and then each half that is zipped apart ends up being the template to fill in, so that you end up with 2. There's an enzyme called DNA polymerase. The ligand splices together, blah blah blah. All we really need out of this is that that you end up with 2 DNA molecules formed from the original. So it ends up looking like this. Now the nucleotides, a DNA strand is a series of these chemicals called nucleotides, A's and C's and T's and G's, so adenine, thymine, cytosine, and guanine, and they're arranged in a specific order. And the order is what matters. Now, A's have a special bonding site that will only bond with T's, and C's will only bond with G's. So what happens is that the two strands unzip like a zipper, and then on the template of the unzipped strand, free nucleotides bond and fill in and lead to two new strands of double nucleotides that are exactly the same as the old one. So, so that it ends up being the same. That's what happens during S. Now, there are 46 strands of chromatin. Now, when we enter the mitotic phase of the cell life cycle, if it's going to happen, it doesn't happen with nerves, skeletal muscle, cardiac muscle, but if it's going to happen, then we go into this mitotic phase. And this becomes mitosis and cytokinesis. So mitosis is nuclear division, and there's 4 stages of it: prophase, metaphase, anaphase, and telophase. I, in class, talked about this already, but I'll talk about it again. This is the order that they happen. We'll go all the way down here to prophase. So, in prophase, what happens is this. This is preparing. Pro means first or pre. What happens is the chromosomes, the chromatin starts to wind up and onto its spools and become chromosomes. And we end up with 46 chromosomes. We end up with 23 pairs. And basically each chromosome is like a recipe book. It's like a cookbook. You get one cookbook for certain proteins from your mother, and you get one cookbook for the same proteins for your father. Which cookbook you express, which ones you use, which recipes you use, is dependent on which ones are dominant, which ones are recessive. But we're not going to get into the genetics of it all. But nevertheless, these things condense. The chromatin basically winds up and becomes a less of a tangled mess and more like 46 spools of thread, 23 pairs. The centriomes separate and they migrate to opposite poles and spindles form, and those are really kind of the push-pull rods that move crap around. All of this happens in prophase. The other thing that happens in prophase is that the nuclear envelope starts to break up. So the lipid bilayer of the nuclear envelope starts to disintegrate, because now the chromosomes, once they're formed, don't need to be protected the same way the chromatin does. Now, there are tubules that join the centromeres, the central part of the chromosome, to the centrosomes. And they start to move these chromosomes apart or around. So what happens is these things move, the nuclear envelope disappears, the chromosomes start to form in the chromatin. Late in prophase, you end up with this. And the chromosomes start to kind of pair up and get moved around where the nucleus was. The nucleus is now gone. In metaphase, which is the second phase of mitosis, nuclear division. The chromosomes align along the middle of the cell. That's why it's called metaphase, kind of halfway between the two poles. We call this the metaphase plate. It's somewhat irrelevant, just that they line up in the middle of the cell. So they end up like this. All the chromosomes end up lined up across the cell. This is an actual photograph of chromosomes lining up in metaphase. All the little microtubules here in yellow go to the centrioles, and that's what causes the lineup to happen. The next thing that happens is anaphase. Ana means apart. So this is a very short little phase where the chromosomes, which have the two strands of chromatin, so the replicated DNA is still packaged together, and it's joined together at a button in the center called the centromere. And what happens at anaphase is that the centromere disintegrates and splits, and now the chromosomes split apart. Now, technically speaking, when the chromosomes are still joined at the centromeres, they're called chromatid. They become chromosomes at anaphase. It's a technicality. Nobody's going to correct you if you call them chromosomes during prophase or metaphase. So then the chromosomes start getting pulled towards opposite poles by the little microtubules that are involved with the centrioles. So they end up looking like this. And they kind of look V-shaped just because they're getting pulled through the cytoplasm. The next phase is called telophase. Telophase is when the movement stops, when the chromosomes have reached opposite poles. And it's basically prophase in reverse. So then what happens is the chromosomes start to uncoil and become chromatin again. The nuclear envelope starts to form around and starts to enclose them. The nucleoli reappear, they start to form, and the spindle, the mitotic spindle, disintegrates and is used up. So really, telophase is prophase in reverse. Then, in some cells, we have the cell itself, the cytoplasm, the cell membrane, divides into daughter cells. It starts to happen during anaphase, and basically the stuff is split apart. So the cytoplasm contains organelles and inclusions and all of that stuff. The thing is that it's not equally distributed. It's saying that each nucleus is identical to the original, because now we've paired them up and divided them. But the cytoplasm isn't necessarily equally shared. The cell during G1 will grow the organelles that it needs. So it needs more mitochondria. So say one daughter cell gets more mitochondria than the other, then the other one just grows more. Organelles are reproducible. They are, you can form them. So cytokinesis really gets fully underway during telophase, and then it finishes after telophase is finished. And you end up with two daughter cells with identical nuclei. So what happens here? how does the cell know when it's time to happen? Well, they grow to the certain point where the volume, which is expanding cubed, like to the third power, outgrows the ability of the cell membrane, which is expanding squared to the second power, and there's inadequate exchange, so you have to divide it. There are other things, different hormones, that growth factors, etc., that make it happen. There are growth inhibiting factors that stop it. Contact inhibition is when there's enough. So if you cut yourself and your epithelial cells are not in contact with one another, they will reproduce rapidly until that cut heals over, and then when the two sides of the cut are in contact with one another, the rate of mitosis slows right down. So fundamentally, that's what's going on, contact inhibition. So that's a brief look at the cell life cycle, interphase and mitotic phase.